Alright, hello Bio, this is Mr. B, continuing on with Chapter 11 in our chapter about genetics, and today we're going to talk about probability and Punnett squares. So we'll get right into it. So the first thing we're going to look at is how do geneticists use the principles of probability, and we're going to need to get a definition of probability before we can talk about what it means. So the likelihood that a particular event will occur is called probability. So that's simply our definition right there. The likelihood that a particular event will occur is called probability. And the principles of probability can be used to predict the outcomes of genetic crosses. Now that's the most important word there, is predict. So it's not always going to be 100% accurate, the same thing every single time. It's just kind of us giving away, gives us a way to give kind of a general outlook on what's going to happen so we can predict what might happen, not say, oh, this is what's going to happen every time. So that's how we use probability in the terms of genetics. We're going to be able to kind of map out what we think is going to happen, and we can kind of go from there. And a tool that we use to kind of look at the probability of how things are happening here is called the Punnett square. So that's going to be important to know for this section, and this whole chapter is called Punnett squares. And what Punnett squares are is they are a tool that uses the gene combinations that might result from a genetic cross, and they can be determined by drawing a diagram known as a Punnett square. So our Punnett squares help us determine the genetic crosses that could be possible, and they're used to predict and compare the genetic variations that will result from a cross. So it's again using probability because they're not always 100% correct, but it's us giving our best guess at what might happen. So we're going to take a look at an example of what a Punnett square will look like, and just some general rules when we write a Punnett square. A capital letter will represent our dominant allele for a trait, and in this case it is tall, so we're going to use a big T for tall, and a lowercase letter represents the recessive allele for short, so we have little t for short. So we're not using T and S, we always are going to use a capital letter for the dominant trait, the lowercase letter for the recessive trait. So that's our first rule here. And then our next is that the gametes that are produced by each F1 parent are shown along the top and the left side. So our gene combinations of our parents are on the top and left side. So we have up on the top, there's one parent, up on the left, we have the other parent. So that is our next rule here, and as you can see, we have our gene combinations, our possibilities here, written right there in these boxes. So we have big T, little t, big T, little t. So those are all the possible gene combinations that come along from segregation. And then our possible gene combinations for the F2 offspring are going to appear in our 4 boxes. So we have all of our combinations. As you can see here, we have a big T and a big T, so this box right here gets 2 big T's. We have big T and little t, so we have big T, little t. Big T, little t, big T, little t. Little t, little t, little t, little t. So that's how this works. You just kind of go over and you match them up. It's pretty simple when you only have 2 traits. It's kind of self-explanatory. So that's what a Punnett square looks like. We'll do some practice with Punnett squares in class. And organisms, so that's it for Punnett squares for now. We're going to take a look at some important definitions that you should have already defined at one point on our introductory activity. And so we're going to look at the fact that we have something called homozygous versus heterozygous to start. So organisms that have 2 identical alleles for a particular trait are said to be homozygous. So if we go back to our picture here, we have big T, big T, big T, and little t, little t. Those are our only 2 that are homozygous. One's homozygous for the dominant trait because it has 2 big T's, and one is homozygous for the recessive trait because it has 2 little t's. And then our next definition is organisms that have 2 different alleles for the same trait are heterozygous. So if we go back our other 2, we can see I have big T, little t, big T, little t. Those are heterozygous because they have 1 of each. And then some just kind of things about the homozygous versus heterozygous. Homozygous organisms are true breeding for a particular trait, and heterozygous organisms are hybrid for a particular trait. So that's just kind of the same way of saying that they have 2 identical alleles, so they're true breeding, and heterozygous, they're hybrid, meaning they can pass on either trait. And then some more definitions. All of the plants have the same phenotype, or all of the tall plants have the same phenotype. This is going back to our example. or physical characteristics. So when we see the word phenotype, that means physical characteristics, and that's kind of if we have pH at the beginning of both those words, phenotype, physical characteristics. And then, however, our tall plants do not have the same genotype, being genetic makeup. So if we go back and we look at our example here again, we have big T, that's the and then big T, little t. They're both tall, they both have the same physical characteristics, phenotype, but they do not have the same genotype because one has a little t. And so then kind of some more, and then 1/3 of the tall plants are TT, while the 2/3 of the tall plants are big T, little t. That's what we just said. So these plants have different genotypes. You can see big T, big T, little t, but they have the same phenotype. And then here's just kind of our probability. So 1/4 of the F2 plants have 2 alleles for tallness, half have the allele for tall and short, and 1/4 have 2 alleles for short. So that's pretty simple to see there. We have 4 boxes, and we just look at the possibilities that we have there. And then because the allele for tallness is dominant over the allele for shortness, 3/4 of the F2 plant should be tall, and this ratio is going to be 3:1. And this predicted ratio showed up in Mendel's experiments, indicating that segregation did occur. So these Punnett squares kind of validate the fact that segregation did occur. And then just kind of some closing thoughts here. Probabilities predict the average outcome of a large number of events, and this most important part here, they cannot predict the precise outcome of an individual event. It's just our way to kind of guess almost, or an educated guess to make a hypothesis. And in genetics, the larger the number of offspring, the closer the resulting numbers will get to the expected values. So that's it for this video. Let me know if you have any questions.